Hey there, welcome to Stork Science World. I'm Jamie, and today we're diving into some fascinating research in the field of cancer immunotherapy. This podcast is produced by Stork, a research productivity software that's revolutionizing how scientists work. Alex, I'm really excited about today's topic.
Absolutely, Jamie. Today we're looking at a groundbreaking study about gastric cancer immunotherapy. It was just published in Nature Communications, and it addresses a major challenge in cancer treatment: why some patients respond to immunotherapy while others don't. And let me tell you, this one has some really promising findings.
You know, this is particularly important because gastric cancer is one of the leading causes of cancer-related deaths globally. And while immunotherapy has shown amazing results in some cancers, it's been a bit of a mixed bag for gastric cancer patients.
Right, and that's the key issue this study addresses. So, for some context, immunotherapy drugs called immune checkpoint inhibitors, or ICIs, work incredibly well for a small subset of gastric cancers that are classified as microsatellite instability high, or MSIH. These patients have about a 45 to 60% response rate, which is pretty impressive.
But the problem is that those MSIH cancers only represent about 10 to 15% of gastric cancers, right? The vast majority, over 90%, are what's called microsatellite-stable, or MSS, and those patients have a much lower response rate to immunotherapy, only around 10%.
Exactly. And that's where this research team from China Medical University made their breakthrough. They asked, what if we could identify which MSS gastric cancer patients would respond to immunotherapy? And even better, what if we could find ways to make more patients responsive to these treatments?
So how did they approach this problem? It sounds like a needle in a haystack situation.
Well, they used some pretty sophisticated machine learning techniques. They developed what's called a causative unlabeled learning model, or PUL for short. Basically, they trained an algorithm to identify patterns in MSS tumors that resembled the immune characteristics of MSIH tumors that typically respond well to immunotherapy.
That's fascinating. So they were essentially teaching a computer to recognize which MSS tumors might behave more like MSIH tumors when it comes to immunotherapy response. How accurate was their model?
Incredibly accurate, actually. Their model achieved an area under the curve of 0.924, which is, well, that's about as good as it gets in predictive medicine. They could effectively identify which MSS patients would respond to immunotherapy with remarkable precision.
Okay, so they created this super accurate model to predict which patients would respond. But what did they actually discover about these tumors? What makes some MSS gastric cancers responsive, while others aren't?
That's where it gets really interesting. They found that the key factor was a specific type of immune cell called TBET plus CD8 plus T cells. These are a special subset of CD8 plus T cells that express a transcription factor called TBET.
And what makes these TBET plus CD8 plus T cells so special compared to regular CD8 plus T cells?
So these TBET plus CD8 plus T cells have two really important properties. First, they're much better at infiltrating deep into tumors. The researchers showed that while regular T cells often get stuck at the edges of tumors, these TBET plus CD8 plus T cells can penetrate right into the tumor core.
That's crucial because immunotherapy can only work if immune cells actually reach the cancer cells, right?
Exactly. And second, these TBET plus CD8 plus T cells are much more effective at killing cancer cells. They produce higher levels of cytotoxic molecules like granzyme B and interferon gamma. The researchers showed that patients with more of these TBET plus CD8 plus T cells in their tumors had significantly better survival. We're talking 77 months median overall survival versus just 24 months.
Wow, that's a huge difference in survival. So basically, these special T cells can both reach the cancer better and kill it more effectively. Did the researchers look at what happens at the molecular level? How do these cells actually interact with the tumor?
They did, and they discovered something really interesting:
a positive feedback loop between these T-bet plus T cells and the tumor cells. When T-bet plus T cells infiltrate the tumor, they release interferon gamma, which causes nearby tumor cells to express PD-L1 and produce chemokines called CXCL9, 10, and 11.
And those chemokines, they're like chemical signals that attract more T cells, right?
Exactly, so it creates this virtuous cycle where T-bet plus T cells enter the tumor, cause tumor cells to release signals that attract more T-bet plus T cells, and so on. The researchers could actually see these areas they called T-bet plus PD-L1 plus niches in the tumors of patients who responded to immunotherapy.
And I'm guessing the PD-L1 expression is particularly important here, since that's what the immunotherapy drugs target, right?
You've got it. PD-L1 on tumor cells binds to PD-1 on T cells, which normally suppresses the immune response. The immunotherapy drugs, PD-1 inhibitors, block this interaction, allowing the T cells to remain active and kill the cancer cells. So these niches create the perfect conditions for immunotherapy to work.
So, this explains why only some MSS gastric cancers respond to immunotherapy. They're the ones with enough of these T-bet plus CD8 plus T cells. But here's what I'm really wondering. Did the researchers find any way to increase these special T cells in patients who don't have enough of them naturally?
That's the most exciting part of the study. They actually did experiments where they transferred T-bet plus CD8 plus T cells into mice with gastric tumors that were previously unresponsive to immunotherapy. And guess what happened?
Don't leave me hanging, Alex. What happened?
The tumors that were previously resistant to immunotherapy became responsive. The transferred T-bet plus CD8 plus T cells infiltrated the tumors, started that positive feedback loop we talked about, and when combined with PD-1 inhibitors, they significantly reduced tumor growth. It's like they transformed a cold tumor into a hot one that immunotherapy could then target.
That's incredible. So this isn't just about predicting who will respond to treatment. It's potentially a way to make non-responders into responders. I can see why this research is so significant.
Absolutely. And they also found some interesting genetic factors. For example, patients with mutations in a gene called PIK3CA were much more likely to respond to immunotherapy. In their validation cohort, 75% of MSS patients with PIK3CA mutations responded to immunotherapy, compared to only about 15% of those without the mutation.
So to sum up, this study has really given us two major advances: a way to predict which gastric cancer patients will respond to immunotherapy with incredible accuracy, and a potential therapeutic strategy to help patients who wouldn't normally respond.
That's right. And while this research focused on gastric cancer, these findings about T-bet plus CD8 plus T cells could potentially apply to other cancer types too. I think we're going to see a lot more research building on these discoveries.
Well, that wraps up today's discussion on this fascinating research. Just to remind our listeners, this study titled T-bet plus CD8 plus T cells govern anti-PD-1 responses in microsatellite-stable gastric cancers was published in Nature Communications. And as always, we want to thank Stork for producing this podcast and for their continued support of scientific communication.
Thanks for listening, everyone. If you enjoyed this episode, please subscribe and join us next time on Stork Science World, where we'll be exploring more cutting-edge research that's changing our understanding of science and medicine.